So the next talk is the hype and the hope in gene therapy trials. We're actually going to have two speakers here share this talk. One is Dr. Sam Frank, who's an associate professor in neurology at Harvard Medical School, also director of our Huntington's Disease Center of Excellence. But he also has extensive experience in both clinical care and research on patients with Parkinson's disease. And Dr. Veronique Vanderhorst is an assistant professor of neurology at Harvard Medical School, and she leads preclinical and translational research on gene therapy studies here at Beth Israel Deaconess Medical Center. Who's first here is Dr. Frank. Okay.
Well, thank you for having us, and this isn't going to be as much of a debate, maybe a little questions back and forth at the end, but I think that what we wanted to kind of convey was some of what's been done already in terms of gene therapy or gene-based treatments, not only in Parkinson's disease, but some related disorders that we can apply to Parkinson's disease, and then what's going to be potentially used in the future. And so I got the short straw, and have to talk about what's been done before and provide more on the hope side rather, and I should say the hype side rather than the hope side. But in full disclosure, I think Dr. Vanderhorst and I both are really hopeful in terms of the potential use of these therapies in Parkinson's disease. And so what I'm going to do is talk about some of the past clinical trials that have gone on. These are actually still going on in terms of collection of data in Parkinson's disease. And then Dr. Vanderhorst is going to talk about some of the research that's going on for gene therapy related to Parkinson's disease, and then we'll have a little bit of discussion and, of course, answer some questions. One question that comes up just about every clinic is, is Parkinson's genetic? My grandfather had it, or my sister has it, and this is from Prevention Genetics. It's a commercial lab, and you can send off genes that increase your risk for Parkinson's disease in various patterns of inheritance. And so sometimes it is in every generation, sometimes it's only in men, sometimes it just increases your susceptibility, and so this is something that we can send off. Keeping that in mind, more than 90% of cases of Parkinson's disease are not genetic in nature. And so we are still looking for genes that increase risk and that may be able to be the target for future clinical trials. But in general, Parkinson's disease is not a genetic disease. So it's only about 5 to 10% of cases that when we go looking for a gene, that we find one. That being said, interventions can still be gene-based because we know which proteins may be involved in Parkinson's disease. I want to spend a little time on this slide. I am not going to get up here and talk about a lot of data. I just want to be clear about that. But I think that it's important to see what's been tried. And I put this up here when I was doing my fellowship at the University of Rochester starting in 2002. I was involved in the Parkinson's disease gene therapy study group. And so that was in 2002, and we are just now getting to some of those interventions that are being looked at. So this is an ongoing and longer process. The first three are really for symptom control, so it is not to change the course of the disease, but what can we do to improve symptomatic therapy and control? And I hope that you can see, at least from the first one, the adeno-associated virus. There was an adenovirus that was used in another disease, but it caused a tremendous inflammation in the body, and in fact, there was some deaths related to that. And so all gene-based therapies were actually put on hold for about a decade. And we found that the AAV, the adeno-associated virus, is actually a much safer virus. And this was one of the first studies that was published in 2007, that first one, and it showed that it was safe. There's still ongoing collection of those participants in that study, and 4-year data has actually been collected and has been published. In 2017, it was published. The same type of virus, AAV, was used to deliver AADC. AADC is one of the enzymes that converts levodopa to dopamine in the brain. So to make that more efficient, essentially. And again, this has been going on, and some of the early work was published in 2009, 2008 from some of the theoretical and methods work. And there are commercial entities that have picked this up and are moving this forward. One is a study that we're doing at BI with Voyager Therapeutics. But again, it's for symptom control and using the same virus. And then Prosavin is the name of the agent that one company put through. Actually, I pulled it up, and it said it's based on equine infectious anemia, virus encoding AADC, and cyclohydrolase 1. I'd much rather say Prosavin. So that's what that agent is, and it's a different vector that's being used. It's a lentivirus. And so just to have one bit of reality in terms of gene-based therapies, most of what's been studied is really for symptomatic control and various different viruses. We're still looking for the exact correct type of virus and agent to deliver the genes. There was one study of potentially disease-modifying called SARA-120, and this was, again, an adeno-associated virus that delivered a gene that interferes or enhances nurturin, which is a growth factor in the brain. And unfortunately, this was negative in terms of not causing any changes from a disease modification perspective, but that's an ongoing study and continues to have publications. The initial results were published in 2008, and there was a recent publication in 2016 that really confirmed that. So I think we're looking for what's the right virus and what's the right purpose of the gene-based therapies. And I hope you got the idea from Dr. Fox's talk that Parkinson's disease is not just one problem in one tiny little area in the substantia nigra that we often talk about when it comes to dopamine production in the brain. It's a whole circuit, and much of the brain is involved. And so when you look at what's been studied, the subthalamic nucleus, which is one of the targets for deep brain stimulation, the putamen, which can be a target for deep brain stimulation and focused ultrasound, and the substantia nigra, which we really haven't messed with from other aspects. But we don't know what the right target is in terms of inserting genes and messing around with the genes in the area. So we're still looking for what's the right target. Now, I also want to spend just a minute on how these different interventions have been studied. I took the opportunity to go to my grandfather's anatomy book from 1933 to pull up a skull drawing. And the reason I do that is because we have to drill through both layers of the skull to insert a catheter and then infuse the fluid that contains the gene and virus. When we talk about phase 1 studies, that's first in human. So that is not just in mice, but we're moving toward humans. And the primary purpose of phase 1 studies is safety. Is inserting a gene into the brain, is it safe to do that? And I think that's obviously a critical importance to answer that question when we are putting these agents into humans. And what's the right dose? We don't want to overwhelm the brain, and we want to give enough so that the gene gets inserted and is able to do its job. So that's the phase 1. Phase 2 there's a double-blind sham surgery controlled. And so what a sham surgery is just taking out the outer part of the skull. There's still the inner part that is intact, but you go to the operating room, they make an incision, they take out the outer layer of the skull, and then we sit there for the amount of time that it would have taken to infuse the drug. Now, this is where it gets a little tricky. If you look at past surgical interventions, not necessarily gene-based therapies, but past surgical interventions for Parkinson's disease, it's actually quite important to have a sham surgery control. There was a study looking at adrenal autotransplant where we would take cells from the person's adrenal gland that sits above the kidney and implant that in the brain because both produce dopamine. And it took almost 800 people to have that surgery before we figured out it doesn't work. Then there were different cells, embryonic cells that would go on to produce dopamine that we put into the brain. We had a few hundred people that had that, and we said, you know what? Let's not reinvent the wheel. Let's not repeat history. Let's have a sham surgery controlled. And so there were 2 studies funded by the National Institutes of Health to look at whether those cells that we're going to develop into cells to produce dopamine would work, and it turns out that it really didn't in the vast majority of patients, and in fact, there were side effects. And so this is really the standard now moving forward, is to have sham surgery control, and it's essentially a minor orthopedic procedure if you think about it. It's just removing one aspect of the bone. And some of the studies that were not phase 1, but we've gone on to phase 2, testing some of these agents. That's how we've really figured out that the surgery itself did not cause any problems, and there was no benefit either. And then open label extension, and that's what's going on now. Once you put a gene into someone through using a virus, we assume that it stays there, and there are certain measures so that we can look at that, and in fact, it does in the vast majority of what we've done. And in general, this is, again, I'm not going to show the data of the couple of dozen studies that have been done so far. In general, these interventions are safe. Of course, we do run into some adverse effects. Headache and bleeding around the brain are the most common potential adverse effects. And I think that it's become clear, even though it's just been one agent, that that one agent is not effective for disease modification. But it may be effective for symptomatic relief, and that's what we're about to do, is to test that in a larger, longer study, and may reduce the need for more levodopa. I think that we need to think about gene-based therapies in Parkinson's disease because there have been some successes in other disease states. And I just want to show a little bit of data for some of those successes. And hopefully we can take what we've learned from other disease states and apply it to Parkinson's disease. The first is nusinersen, which is a drug that's called an antisense oligonucleotide. And so just to refresh your memory in terms of genetics, DNA is the blueprint for proteins, and RNA is in between. So it's DNA that gets converted to RNA, and RNA is basically there's different kinds of RNAs, but they help to build the proteins. And so there's an antisense oligonucleotide, antisense meaning that it complements either the RNA or the DNA, and oligo meaning few nucleotides, which is we're talking about RNA and DNA. So it's a human-made section of RNA or DNA that interferes with the process or augments a process depending on what we want. Now, spinal muscular atrophy is a really bad disease, especially type 1. 90% of kids actually die by the age of 2 because of lack of muscle control, and they stop breathing because they don't have the strength. And this is the open label extension study of kids that got this drug. And the fact that we're talking about the 6-minute walk test is really remarkable because these kids never walked before. So the fact that we can even test walking is amazing. So these are kids that we give a shot in their back and deliver this ASO into the spinal fluid. It was initially once a month. It's now down to once every 4 to 6 months, and it interferes with the process. And so they make more of the protein that they need and can actually develop relatively normally, which is pretty remarkable. And these kids are now 7, 8, 9 years old, and they never would have made it to 2 otherwise. So I think that that's one of the most exciting and promising interventions that we've seen. A similar technology is being applied to Huntington's disease. Now, Huntington's disease is a genetic disease. You just need one copy to inherit this disease, and it's purely genetic. That's the only way that you're going to get Huntington's disease. But it impacts very similar circuits and structures in the brain as Parkinson's disease. So I think what's going on in Huntington's disease very much can be applied to Parkinson's. And it's similar technology in terms of antisense oligonucleotide therapy, interfering with the development of an abnormal and destructive protein in this case. And this is 46 people that were given, again, a shot to the back once a month. And this is the phase 1. And so there's different doses. And you can see the higher the dose, the lower the amount of abnormal destructive protein. So we are in the process of testing this in a larger cohort of people around the world. again, it's something to pay attention to in terms of gene-based therapies, and ASOs are being developed for Parkinson's disease as well. There's no data to show you, but I can tell you that there are multiple companies that are out there looking at how can we use an ASO to interfere with the production of abnormal alpha-synuclein. I would be remiss if I didn't talk about CRISPR. CRISPR is always talked about when it comes to gene-based therapies, and this is hot off the press. It really had been just in animal and cell-based models until very recently, like this week. And this is Vertex, which is in our backyard, and they announced positive safety and efficacy data from 2 patients, so we're not talking about a lot of people, with investigational CRISPR gene editing for hemoglobinopathies. One of them had sickle cell disease, and that person is out in the news and talking. So this is really exciting. CRISPR is gene editing. So what can we do to get into the genome and edit what's going on in there and fix the problem for gene-based disorders? Or is there some way that we can muck around with the DNA and produce some good? I think we have a long ways to go in terms of CRISPR in general, and certainly when it comes to neurologic-based diseases. But I think that it is important to acknowledge that in certain cases, CRISPR is moving into human-based research. Now, all of this comes with a big caveat in terms of that promising work, because those disorders that I've just showed you are based on single gene disorders, which in general Parkinson's is not. And I hope you also see that dopamine is impacted in Parkinson's disease, but there are many transmitters, there are many circuits, there are many structures that are involved. So I think we have a long ways to go in terms of where we're going to put our potential therapies and where we need to test. So I think that I can't say gene therapy for Parkinson's disease, no. I don't think that we can say yes right now either. I think there's a lot of work to be done. And so right now, I think the best that I can say is maybe. And I think that a lot of that work is being done right in our backyard. And with that, I'm going to turn it over to Dr. Vanderhorst to tell us about some of that work that's going on. And we'll take questions at the end after both of our talks. Thank you.
Okay, so that was very exciting. So if this is as negative as you can get it, that's still quite positive. So now it's my turn, and I'm very positive. But gene therapy is not going to make you glow green, okay? So it's a big word, and there is a lot of hype in the word itself. But I hope in this presentation that I can make it clear what it is and actually what it's not. So you may get a little bit better feel of where we are in the field, where we still have a lot of work to do. Now, the way I look at gene therapy is it's a tool. It's just a tool. And you have to learn how to use it in the right way and apply it for the right problem. So you can have a wonderful box of tools, and you have a problem. You need to get a nail into the wall. And, well, if you take the screwdriver out, you're going to have a problem because that's not the right tool. And even if you have the most beautiful and well-balanced hammer in your hand, you still need to learn to use that tool. Have you ever seen a toddler kind of hold a hammer and hit its nail or its thumb? So it's very important to use these tools in the right way. And that's where we still have a lot of work to do. So Dr. Frank gave us a little bit of an overview what is in the pipeline that's relevant with respect to gene therapy in patients with Parkinson's disease. And much of the approaches are focused on dopamine. And that's a little bit of a problem because, as we know, a lot of the circuits in Parkinson's disease involve more than the dopamine system only. And it's all how these circuits work together. It's eventually what makes the symptoms, but it's also what makes some of the systems really strong. So there is a balance between circuits that are never affected and circuits that are affected. Especially the non-dopaminergic circuits are the ones that are important for walking, balance, and gait. And would it be possible to use gene therapy approaches to actually focus on those non-dopamine circuits to help those really challenging symptoms? So that's in the end what we would like to do. Now, these circuits, just to illustrate some of the other circuits than the dopamine circuits, these circuits are affected by the pathology that's common in Parkinson's disease, is alpha-synuclein, and that makes those clusters there, these little Lewy bodies, and they're indicated in brown. Now, if we, for example, look in the brainstem, that's an area that's extremely important for balance and posture and walking. And if we look at that, this is a reconstruction of a human brainstem. You see the brown areas, that's the substantia nigra, but there are also clusters of brown throughout the rest of the brainstem. And those are the areas that are so important for walking and gait. And many of these areas, they actually connect to the spinal cord. So going back to Dr. Fox's talk with the spinal stimulation, most likely some of those effects could potentially target those tracts that go down into the spinal cord. Now, can we use gene therapy to target these non-dopamine circuits? Can we do that? Well, let's think about that. And what I'd like to do is actually take you through a series of experiments or an approach that we have developed here at BIDMC with the support of the community, so patients and their caregivers. And that's a project that's been moving forward. So the idea is that in Parkinson's disease, there is loss of nerve cells in the brain. And that makes that circuits get out of balance. And because of that, that's when these symptoms arise. So these symptoms are slowness, stiffness, and shakiness. So this is as if the brake has been applied to these circuits in the brain. Now, if that's the problem, why don't we come up with a tool so that we can release that brake, so that people can move freely again? Isn't that what you do with your car? Okay, you go to a mechanic and you fix the whole problem. Now the problem is, with respect to the human brain, that we didn't design the brain ourselves. So we have designed cars over time, and there are mechanics that know enough about cars to be able to fix it. They know where all the circuits are in the car. These brains, they have evolved over millions of years, and we understand some of it, but we don't understand all of the circuits and the neurotransmitters. We just don't understand them well enough. So there are gaps in our knowledge that makes it very hard to apply a wonderful tool in a smart way. So we still have some work to do there. Now, even though mice are not people, we can use mice to really develop our ideas and develop approaches very fast to screen out failures and then only move forward with the most promising tools and approaches in people. And that step can be challenging as well. So do we know where all the breaks are and where all the symptoms that occur in Parkinson's that we cannot treat with levodopa, where they're coming from? No, they're quite yet there, but there are some circuits where we do know how they function and where they're sitting. And one of them is the subthalamic nucleus. So Dr. Fox talked about that. Dr. Frank talked about that. So it's sitting very deep in the brain. And can we use a gene therapy approach to change that circuit? It's a circuit that's non-dopaminergic. And if it works for this circuit, we could potentially apply it for many of the other circuits, for example, the ones in the brainstem. So can we dampen the activity that's being done with deep brain stimulation already? But it would be nice if we could do that without hardware. And if we could maybe get rid of some of the side effects by targeting the gene therapy to certain subtypes of cells. because there are good cells and there are bad cells to stimulate. And there are other advantages to an approach like that as well, especially in areas where there are a lot of fiber tracts that may get stimulated by the electrical stimuli, and you don't want these fiber tracts to be stimulated at all. So how does that work? So we have our circuit, so we have our circuit node, that's the subthalamic nucleus. We have our symptoms to fix, that's slowness and stiffness and shakiness. Now, what are these gene therapy tools? So what these gene therapy tools entail is we can build in genes into the nervous system. And that can be genes that promote a certain neurotrophic factor, which has been done already in people, and it wasn't that effective. But we can do this also by incorporating mutated receptors or ion channels. Ion channels are receptors that normally don't occur in our brain. So we can build them into a particular circuit, and then we want to control them. We want to control them in such a way that these receptors are not just going to sit there and make more and more, but we want to be able to turn them on and off when we want it, and we want to be able to dial them up and down as much as is needed. So we want this gene therapy to be not an on and off type of thing, but we want this to be modifiable. And that's where the chemo part comes in, the drug part comes in. So we can use a drug that doesn't do anything anywhere in the brain, but sits particularly on those receptors and ion channels that we have built into the brain. And then we can dial that up and down. Now, you've heard Dr. Frank talk about those viral vectors. Now, what are those? It sounds scary actually, because the word virus, isn't that bad? Well, these viral vectors are derived from viruses, but the bad parts have been removed. So these viral vectors cannot replicate themselves. They cannot make more and more and more of themselves. And the bad message has been taken out of these viruses, and it has been replaced by the genes of, say, the neurotrophic factor, or in our case, those receptor channels. So what happens is the viral vector then sticks to a nerve cell, gets in there, the gene is inserted in the cell, and then the cell is going to make the receptor or the ion channel, and that cell is going to sit there. And in our case, nothing is going to happen yet. So these cells are just happy as they normally are, and they may be working too hard or not hard enough, but then when we give the drug, then we can turn this circuit up and down. So that's indicated on the, oops, where is the pointer? That's indicated here. This is the circuit. You bring in the viral vector, and that's with an injection, so it's still a surgery. And then nothing is happening unless you give the drug. You turn the circuit down in this case, and then once the drug is washed out, the whole circuit is back to its baseline. So this gives enormous control. Now, here it gets to the details. So why do certain studies or certain trials not work, and sometimes they do work? So that has to do with the types of these viral vectors. So they come in a lot of codes, and those codes are labeled with numbers. And some of those numbers, if you have, say, for example, a 10, you get a really, really large area where the virus spreads. If you use a 2, the virus doesn't spread much at all. So depending on your target, you want to have a virus with a code that spreads a lot or doesn't spread. Now, the other important part is that depending on the code, you can keep the virus at the site, or you can send it along the fiber that the nerve cell uses and have it go in one or the other direction. This is also really important depending on the circuit you're targeting. So if you choose the wrong number, everything is fine, but you just don't have the correct delivery of the gene at the site you wanted to do. And then there are other tricks that we're using, and they're also very important. We can select which cell types to target a viral vector to. So we've done a lot of proof of principle experiments in mice, not 3 mice, many groups. It's very reproducible. And what we do in those mice, we make the mice Parkinsonian. So you see the substantia nigra here in brown, and on the other side it's white. So there are no substantia nigra cells that make dopamine. Then the virus is inserted here. It's labeled in black, and it's transfecting the nerve cells in the subthalamic nucleus. And then what we see when we test how these mice walk, they walk much faster here in purple and much faster here in blue than when they don't have this circuit activated. So we dampen the activity of that subthalamic area. And it's reversible, so we can move it up and we can move it down even before they're Parkinsonian, and it works even better after they're Parkinsonian. And now the nice part is there is a dose response as well. So you see this line here. These are the different doses from low to high, and the more we turn the circuit off, the better these mice are doing. Actually, you say better. In athletics, this would be called cheating because we turn this circuit down way too much. So these mice are actually moving much faster than they would do at baseline. So they only need a very small dose to get a physiologic response. And again, this is completely reversible. I think the screen width is a little bit too wide for what I made the presentation at. So the mouse that I'm going to show is a little bit stretched out for that reason. So that's a mouse, that's Parkinsonian. And it needs a little bit of cueing on the walkway. So this is how we measure its gait, and which is actually a metric that we can translate to people. So that's not an issue at all. And now we've turned the circuit down, so the mouse is much faster, and it has a more of a swing. This is one of the medium doses, so you may say, well, maybe this mouse is a little bit too swingy here. That's exactly what it is, because this is one of the slightly higher doses that we gave. But this mouse is doing everything normally. It's doing it doesn't show any side effects in these tests. So that's very promising. But now you just listen to Dr. Fox, and he was like, well, but you saw his mouse brains, they're so small. Yeah, and he's right. Okay, they are really, really very tiny, and that's our brain, much bigger. So we do have a challenge there, and it's something we have to take into account. So that's why we have been, in parallel to the mouse studies and the rat studies, we have been piloting this approach in a primate brain as well. So these monkeys, they go into an MRI scanner to get the viral vector injected, and there is planning similar to that, how that occurs in people. And this is the intraoperative MRI. So during the surgeries, these monkeys undergo another MRI. These monkeys cannot be awake, so we cannot record these cells because they don't like to cooperate as much. You can imagine if you have to be awake during surgery, that's very difficult for patients already, and the monkeys would not understand that. So what we've been seeing after delivering this same viral vector that you've seen in mice, once we dose the chemogenetic drug appropriately, the monkeys start using their affected hands. So that's very promising. And very important for safety, we're now looking at the brains of these monkeys to see whether the virus was safe. Is there any inflammation? Is there any reactivity? We haven't seen that so far, but we have to still do extra stains there. So we're moving in that direction. So this is an example of how a gene therapy approach can be used Not to fix all genes, because that wouldn't work in Parkinson's disease, because in the majority of patients, it's not one gene that needs to be fixed, but it's a combination of a little bit of this and a little bit of that. But we can use these gene therapy approaches to target very specific circuits. We need to know where the circuits are sitting and how they're working, otherwise we use the tool in the wrong way at the wrong site. And that's very important. So that's really crucial for success. Now, what are actually those requirements for success? So if we really want to make optimal use of these tools and get them to apply for problems where it really, really matters, of course, the approach has to be safe, but it also has to work. So if it works and it's not safe, it's not going to work. If it's safe, that's wonderful, and you can feel really good about how it technically looks, but if it really doesn't work, it's not worth it. And there oftentimes is a little bit of a balance, because as we know, even standard medications come with a little bit of a risk. So it's really, how does the benefit weigh against the risk in itself? Now, and there are a lot of requirements. Okay, a lot of requirements, and that's just the way it is. So to do this right, we got to make sure that we check all of those boxes. And the gene therapy, and I think that's a message that's very clear, it's not going to fix all of the problems immediately, at least at this point. That's not what it looks like in Parkinson's disease, again, because there is not one single gene that can do that. And maybe we find a gene at some point that can be linked. At that point, we would be ready to really go attack that gene and make use of that. So to end, gene therapy tools, they're tools. I think they are the future. We're not there yet, and we really need to learn how to best use them. So they're new tools, they're very powerful, and we can do a lot of good with them. But if we don't pay attention to the details or take shortcuts, we actually may do harm. So I'm very positive about this, and ending with the same slide that Dr. Frank ended with. This is the trial that where Dr. Frank is actually a site PI, and we're starting this up as well. So it's not only the future, we're actually in it, and doesn't mean it's going to work. So we have to be a little careful there, but we're going to for sure learn a lot more that we can use for future trials. As, for example, we know now that the AAVs, the viral vectors that have been tried already, we know that the AAV types, those are the ones to focus on. So we don't have to spend too much time thinking about other options. So I think we're ready to take questions at this point. Thank you.
Great, thank you very much. And I don't know if we have flyers out on this gene therapy study, but just so you know, we are now one of the IRB approved sites and ready to enroll patients for the AADC gene therapy trial. There's a lot to know about it. For anyone who's interested, you can ask any of us and we can tell you more. Here's one important question somebody asked. So if only maybe 10% of Parkinson's is genetic, you know, does gene therapy matter for the rest of patients?
So it's true that only about 10% of cases are genetic in origin, but that doesn't mean that we can't intervene from a genetic perspective. And so the gene-based therapies should still be pursued in Parkinson's disease to see if they're safe and effective. I think that what we're really doing is not necessarily getting at the cause of the disease in terms of what we're studying today, but can we intervene in the process to improve the function of the body from a genetic perspective? And I think it's a complex question, but one that we're trying to answer.
Yeah, and then the gene therapy by itself, it's the genes you use to bring into the nervous system. So you don't necessarily have to fix a gene. You can use a gene, insert a gene in a circuit, and use that gene to do something that will improve how the system works. So depending on the definition you use, there is a really wide range of how we can make use of those genes that we can insert into the nervous system to improve function. And that can be the disease process itself, but it can also be a very specific symptom that's linked to a very specific circuit. So there are many, many, many options there. Okay.
Thanks. Before I read the next question, I'm going to ask one question on my own. SMA is an extremely rare disorder, and the gene therapy treatment you mentioned is extraordinarily expensive. What's going to happen if gene therapy is proven to work in Parkinson's, and then there's 1 million patients who might be interested?
Yeah, no, you're exactly right. Spinal muscular atrophy impacts about 2,000 kids per year, probably going to be more than that. And just to give you an idea of what Dr. Simon's talking about in terms of price, the first treatment that kids get is $750,000, and then it's $250,000 for every shot that they get into the back after that. And kids today, we're trying to get away with 2 or 3 per year, but it was originally studied with 1 a month. And so you can imagine it's running, generally speaking, about $1 million a year right now. That pales in comparison to another company that came out with a different approach for this disease, and that it's a one-time treatment, and it's at the cheap price of $2.5 million. So it's the most expensive drug that's out there, but it's a one-time treatment. I think that if you have a small volume, you have to have a big price. I think if you have a lot of volume, I think the price is going to be a lot less. So I think if we can find a gene-based approach that, let's say it's only 1 or 10% of Parkinson's disease patients, we're talking about 100,000 people that may be eligible. So I think that hopefully that volume will be able to keep the price down, but it's still going to be expensive because we're talking about neurosurgery, OR time, the drug itself, and all the evaluations and follow-up that go along with it.
And then another thing to add is that at that point, it becomes really important how we select patients. So we know that not everyone is the same. So the makeup of each and every patient is different. So learning how to select out people who will respond versus people who will need another approach is going to become very, very important as well. And that may help counteract some of those difficulties.
And this question sort of combines a question related to DBS with what you've talked about. A patient asks that typically for cell-based therapies or gene therapy studies, they exclude patients who've had DBS. So if a patient's considering DBS, should they maybe hold off or get genetic testing and wait and see if they're a candidate for a more targeted treatment?
Participation in research is always a personal preference. There are some people that want to go with what's FDA approved and available to help with their symptoms today, and obviously deep brain stimulation would be the choice. If there is someone who is considering participation in a clinical trial, this is one that's available today. There may be others in the future. I think that, again, it's a very personal preference. I think about my brother who builds computers. And he does it just for fun. He's actually a physician. But he said, don't buy a computer for what you think you might need 3 to 5 years from now. Build for what you need today. And so I think that's the same idea. If you really need some type of symptomatic treatment today, you should go for deep brain stimulation. We're still testing whether this is safe and whether it works. But I think that there are some people that really like participating in research. It's fun, and it's really interesting from many perspectives. And so if that's something that you want to pursue, then by all means, consider and get in touch with whoever is running that study. You don't have to have any gene testing whatsoever to participate in clinical trials. And in fact, for this one, it's really for people with run-of-the-mill Parkinson's disease that are interested in participating or maybe deciding between participation in research and deep brain stimulation. So a lot of it comes down to personal preference.
Okay, I think I'm noticing the time now. It's noon, so maybe we'll stop there. Thank you.